All right, everyone, thank you so much for being here today. My name is Brandie Verdone. I am a postdoc in Coppin Collins lab. And today I'm going to talk to you about my, yes, it should be nothing.
Sorry.
It's okay. As long as there's a little one in the corner, right, that's my check mark. The title of my talk today is Of Mice and Men, Strategies of Gaze Coordination. I'd like to thank my fellow SPIO coordinate member Daniel for coming up with that title for me. Okay, so let's begin. So right now, in order to read the sentence on the screen, you're moving your eyes. And it's something that you're probably not thinking about doing, but it's what you're doing to take in the visual information, not only on the screen, but around you. There are people in the room, where you're coming from, where you need to go. And like I said, we are doing this all the time, but we're not necessarily realizing that we're doing it. And not only that, in everyday life, we're not just moving our eyes. Most of the time, our eye movements are accompanied by head movements, as shown to the left, and sometimes even whole body movements take in our visual field. And the sum of our head moving in space and our eye moving in orbit constitutes gaze. Now, coordinated gaze is essential to how we perceive the world. It's essential for stabilizing our visual environment, usually done through done primarily through reflex pathways, and the activity of those reflex pathways. But we also need to be able to redirect our vision in a phenomenon known as saccades, which is an active form of eye movements. And the combination of these two types of eye movements, visual stabilization as well as visual redirection, are what contribute to proper navigation through space. And as you can imagine, improper gaze control can really affect our quality of life. So this GIF on the left is showing, you kind of see that this looks like you're viewing the world through a shaky cell phone camera. That's because that's because what's demonstrated here is a loss of what's called the vestibular ocular reflex, which is what we use to stabilize our vision. On the right, you can see that when things are working intact, we have a smooth ability to follow the person that's running. And so not only is it necessary to have intact reflex pathways in order to stabilize our vision, but we also need to be able to turn off these pathways whenever we want to look somewhere new. Now, paying particular attention to our eye anatomy, we have a region at the back of our eye called the fovea. It's a region of high visual acuity, very condensed region. Now, you, me, and non-human primates are foveates. Now, foveates, having this region of visual acuity at the back of the eye, we can perform what's known as active gaze redirection. So we can look, I can move my eyes, I can look at Josh over here in the corner. And I can do that by performing a directed saccade. I can move my eye, and I don't necessarily need to move my head in order to do that. I can just look over to my left. I can see Josh. However, I and a lot of other people in this room might use mice to study in our laboratories, and mice are what are called aphoveates. Now, aphoveates lack a phobia, they lack that high visual acuity center. So as a result, they're thought to passively redirect their gaze. No region of high visual acuity means that they're going to use their head. I'm going to use my head to look, or they're going to use their head to look at Josh over here in the corner, and their eyes going to come along for the ride. You can think about this sort of like those creepy baby dolls that you might have seen as a kid, where the eye kind of boggles around, comes along with the movement of the head. Now, when we're thinking about humans and primates, and compared to mice, we're thinking about their retinal specialization, whether or not they're foveates or aphoveates, and as a result, what their gaze strategy is going to be. The field pretty much accepts that humans and primates are able to use these voluntary and involuntary gaze strategies to redirect their access to visual gaze, whereas mice are going to use primarily involuntary gaze strategies. But no, I put a question mark there, because despite these seemingly distant or seemingly disparate anatomical features, we do see some conserved circuitry and brain mapping, specifically in a region of the brain called the superior colliculus, which is essential to performing saccade generation. Now, that tells us that it's possible that the strategies of gaze redirection in mice are in fact more like foveates than we previously thought. And so we set out to address this question. And there are two major possibilities based off of what I described. First, what the field pretty much accepts. The mouse uses, the mouse uses the head to redirect their gaze with a reflexive eye movement. So the head moves and the eye comes along for the ride. Or the mouse could be using a more coordinated behavioral strategy to actively redirect their gaze, which is reminiscent of phobiate species, reminiscent of the strategies that you and I use, you and I use. So we thought to address this, and we can do this in the lab by tracking the mouse's head and eye movements to calculate their gaze parameters. So shown here is a mouse that's undergoing a behavioral task where we train them to move their heads between two water spouts for reward. And this is distinct because we're able to really isolate a coordinated behavioral pattern of head and eye movements by training the mouse to perform what's called an active gaze redirection. We have a tiny camera in front of their eye so we can track the eye position, and then we have a potentiometer tracking their head position. And we can compare this active voluntary movement to what we consider a passive movement, which is by definition is head leading, where instead of an animal voluntarily moving their head, I'm moving their head. Now, we found some really striking behavioral parameters when we did these assessments. And the big takeaway is that when it comes to gaze strategies, mice have more in common with phobiates than what we previously understood. First off, you're going to see a lot of these types of graphs or charts in the presentation. These are position traces. So we're looking at the position of the eye with respect to the head and then the overall gaze change. And these tick marks indicate the onset and offset of a given movement. Now, what you see here is that the mice can perform head leading movements where the head moves first. And that's what the literature suggests, right? Not necessarily surprising. But what we also found is that there are cases where the mouse also uses an eye leading gaze strategy, reminiscent of what you and I do, suggesting that animals have a varied strategy of active gaze redirection. In addition, mice also make stereotyped head and eye movements to voluntarily redirect gaze. Now we're looking at this data in terms of velocity. And you can see here that our data collected by the mouse compared to previous work that was done in monkeys showing these stereotyped coordinated behaviors. And in addition to that, we also saw similar behavioral patterns to phobiates, and that the amplitudes of the head and eye increase as gaze amplitude increases. And this is reminiscent of some work that has been published in the monkey literature. So not to go into all of the findings that we found, but just some highlights that we saw that both head and eye position contribute to the overall gaze range. We saw that the speed of head and eye movements contribute to the overall gaze range, maintaining what's called a main sequence relationship. And we also saw that the starting position of the eye determines how much the head and eye need to move to redirect gaze. All of this is reminiscent of what we see in phobiate species, what the literature suggests. But most importantly, and I think this is some of our most exciting findings from this work, is that mice, like phobiates, have distinct strategies for active and passive gaze redirection. So if we compare the mouse moving its head on its own to me moving the mouse, we have varied strategies for active gaze coordination compared with passive. So the mouse isn't just simply moving its head with the eye coming along for the ride, it's showing distinct strategies for how to redirect their axis of visual gaze. And something that was particularly exciting is that when we look at these particular type of passive versus active movement, you might argue that these movements look pretty similar in terms of their trajectories. The eye moves opposite the head and does this quick redirect in both conditions. However, if we calculate how long it takes for that eye to redirect in the active condition, it's much faster than the passive condition, again suggesting that it's a distinct strategy. It's not just a passive phenomenon, it's an active redirection. And so the big take home is that we can start to think of mice as part of an evolutionarily conserved pattern of gaze redirection strategies, not so something that's distinct from other animals. And that actually, in thinking about it this way, we can begin to inform our models of visual vestibular modeling and really bolster up the mouse as a really valuable tool in studying visual vestibular dynamics. And so with that, I would like to thank my current lab, my advisor, Kathy Cullen, all the other Aspire members, and I will take any questions. Thank you.
So in mouse, the coloration of the eye being entirely the same color, whether it's red or black, typically depending on the mouse strain, probably makes it difficult, right? Cats and humans and monkeys, you have a pupil, you can see that moving more easily independently. So when you're doing these monitoring experiments, how do you really accurately measure movement of the eye?
So you can see the whole pupil. They don't necessarily have a distinct iris.
Okay.
So you can still see the pupil movement. And that has a big contrast to the sclera.
Oh.
So yeah, you can definitely make that out, especially with our tiny cameras.
Nice, very cool.
Do you think that there's any that maybe because you see these I guess conserved behaviors in species with and without a phobia, do you think it may have anything to do with the different populations of RGC subtypes? Because for example, you have like your directionally selective ganglion cells, and those help you track like stimulus going across, and so having different populations of different ganglion cells may result in different tracking strategies, because we don't have directionally sensitive cells. You can't track things across like this, so you'll have to have a different gaze strategy.
Yeah, no, that's a very good point. So in addition to having different RGC subpopulations, they have different representations in V1. Specifically, the mouse doesn't have a phobia, it has like a foveal-like specialization that results from their RGC inputs. So I guess to sort of answer your question, I think that even though the RGC subpopulation might vary, the representation still manifests itself in some way. In a way, even though they don't have a phobia, even though they don't have like the same RGC subtype as phobiad species, they still have some sort of phobia-like representation. And so it's sort of like, how are they using that? And that's something that we can explore.
You're sure it's this phobia-like organization rather than a whole other anatomical structure?
So right now it's called phobia-like. So we'll see if that changes.
I wasn't even trying to argue semantics. I just meant, is there a possibility that it could be something else besides that structure in the not the name necessarily?
Potentially, yes. I'm not quite sure what that would be right now. Just because I'm thinking about sort of the distribution of cell types across the fovea and how there's not really like, there's not that peak that you think of when you're thinking about like a cone dense region. I don't know what that would necessarily be in terms of a substitute, but I mean, anything's possible, right?
Are there species that don't even have a phobia-like region that you could maybe test as well, and maybe that will help confirm that that is in fact important?
So you mean in V1 or in the back or in the retina?
Yeah, in the retina.
So they're definitely, yes. The answer is yes.
Okay.
And a lot of people have done that work. Owls, for example, have like a tube instead of a phobia. Cats have a visual streak instead of a phobia. So that so actually a lot of that work exists. The mouse literature, I think in us doing this work, we'll be able to better compare ourselves to the existing literature for animals that don't have a phobia as well. Yeah.
So I know, at least in my research, I mentioned like strain really changes kind of our results, and we have very different research. So maybe this is an absurd question, but
No,
do you think
I know where you're going with it. It's not an absurd question.
I guess which strain do you use, and do you think there's potential for there to be differences in this between strains or even between different rodents?
This is not something I actually have thought about, but I love that you're asking this question, because I feel like back, like in my old work, this is the kind of thing we talk about all the time, like strain specificity. My gut would tell me that, like, if I had to hypothesize, I would say that the strategies should exist across strains. However, I do know that like, for example, so these are done in C57 black 6 mice. If I use a mouse that, I don't know, for example, had like red eyes, I don't know. This is me spitballing.
Yeah.
I mean, I might see something different, I'm not sure. That's not something that we didn't do like strain specificity, but that's definitely something that I feel like I was trained to consider and did not consider this. So I love that you're asking that question. Yeah. Oh, sorry, I'll work my way back.
So the phobia-like region in mice, is generation of your like mature photoreceptors also centered around that region? Because I know like at least in humans, I think other phobias, when you have your phobia, you get this like wave of like, okay, blue cone specification going out, and then you get like red cone specification going out, centered on that region. Do you find, or do you know if there's anything similar in other like visual acuity regions in alternative systems like non-phobias?
That's a great question. To be honest with you, I'm actually not sure, but that's something I definitely need to look into. That's a great question. I'm not sure if anybody else knows. Les?
Brandie, I love your presentation. It's really clear. For not specialists like myself, I really followed it. My question is, Do you think of the head leading and eye leading movements as being independent, or somehow one is reliant on the presence of the other, they're coupled somehow, either in learning or so forth? And another way to ask that question is, if you were to mobilize the head, you see eye leading movements, and if you paralyze the eye, you see head movement.
So that's a great question. I haven't done some of this work, but it's something that I'm interested in exploring down the line, but we do have some hints as to what we think would happen. So there have been some studies where the head is immobilized and the superior clipus is stimulated. And they do see generation of eye movements independent of head movement, which suggests this active gaze phenomena. In terms of whether or not they are linked, I'm not sure if they are dependent on each other per se, but I do think that there is a context dependency, maybe specifically if there's an urgency, if there's predictability, those things might determine which one you use. I guess in me answering it that way, there must be some sort of independent or dependent linkage, if I think about it that way. But I like to think about it as a potential context dependency, for sure. And Shreese, do we have time? I know
You know, if it's too much, we can talk offline, but it's a little out of scope, but I'm just trying to understand. So you started off the talk with phobia, aphobias, phobias. And they have different architectures, the mouse versus the humans and the primates, in that way. But in this context, they have similar behaviors. So what is it? What then, if that's not what human architecture is optimized for, why do we need what is the phobia doing for us that you can't get with mice, that mice can't get, I guess?
Okay. So this is so the reason I'm pausing is this is really deep, and I'm thinking about it from sort of an evolutionary standpoint in terms of what we were. We started off by having a necessity to look at our prey, for example. But then as we became, we went from all fours to upright, we had to look, the movement of our head meant that our visual space was just going to be wobbling around. And so we really needed to come up with a way to not only target our prey, but look or stabilize our vision so we're not constantly walking around nauseous. In terms of what can we do that the mouse can't, I like to think about it, let's definitely talk after this, but I like to think about it in terms of sort of how often are you predator versus how often are you prey? And the mouse has this interesting duality in that it's, it has to hunt for its food, but it's also constantly being hunted, whereas you and I aren't necessarily being hunted in our everyday lives. And so there's sort of this strategy with being able to, or this necessity in being able to take in sort of broad visual information at once, but also be able to focus on your target of interest at the same time, which may not necessitate a phobia in the same way that we need a phobia. I think that's probably, I'll leave it there, but I write about this in my discussion of the paper and we should talk about it afterwards.
I see it's a good answer and that, mice can't thread a needle.
Right.
Right, so, they